I think our understanding is if a cell is driven by these oncogenes, then this is one of the escape mechanisms that can be utilized. And I think one thing we've noticed is as our first-line therapies get better, people are on them for longer, but when they come off them, when it stops working, it's less commonly due to another mutation in that protein that we can target. It's more commonly due to one of those other things. Something else is activated, or it just becomes this dedifferentiated thing on that histologic transformation spectrum. So I think it's happening more possibly because our drugs are getting better, which is kind of a sad irony of it. Although the flip side of that is, as you mentioned, people are living longer. So I think the tumor cells undergoing that selective pressure, remember what happens when we treat someone with one of these really effective therapies is you start with billions of cells to trillions of cells, and then that all shrinks down, 99% die, and then 1% of that very large number or 0.1% is still a lot of cells that have figured out some way to survive that event. And you may not even be able to see it on a scan, but there's probably going to be some residual cells there that have at least learned to exist. They may not be growing, but they've learned to exist. And then it's at some point, something has to change in the cells while that TKI is still there for an evolution bottleneck event.
We hope you liked this video. To support cancer research and get clear expert answers about cancer, follow us and visit CrackingCancer.org to join the Cracking Cancer community.